So the other day I was watching a documentary about buffaloes, right? So basically they live in North America, and you know it was winter time, so the temperatures were like sub-freezing temperatures, and the buffaloes, these magnificent creatures, somehow they survive all that, right? Now the fascinating part is a storm was coming, right? And so the narrator was like, what would you think would happen when a storm in these sub-freezing temperatures already is approaching? Now, a normal human would look at the storm, look at the other direction, and run, looking for shelter, you know, trying to outrun the storm, which he never will, right? The buffalo, on the other hand, it looks straight at the storm and runs through it, collides with it head on, right? So why does it do that? Well, it thinks to itself, instead of running away from the storm and trying to outrun it, well, which will eventually fail, of course, right? And keep running for like 2 weeks and, you know, going through all this miserable process of cold and hail and snow and rain. I'll just run through it, have a couple, maybe 2 or 3 days of miserable weather, and then I'm going to be on the other side of the storm. The storm just moves on, and now I'm happily grazing. The sky's clear, the grass is green, and as they say, the grass is greener on the other side, right? So this is what biochemistry is all about. One of those storms that you absolutely must go through and collide with is NAD. You're going to see it in a lot of enzymes. It's going to be a very important factor in ATP production, right? And once you understand it, it's extremely easy, in fact. Okay, so the mini lectures or the series for the NAD are going to be as follows. The first lecture is going to be an introduction. It is very important. Do not let the weird introduction deceive you. We're going to talk about the definition of it, what it is, not the textbook definition, but what NAD is. So what comes to mind when you think NAD? Visualize it, right? Then we're going to talk about the classification, the forms of NAD, what exactly NAD does, right? And we're going to give you some examples about the general reaction of NAD. Lecture number 2 is going to deal with biosynthesis. Honestly, lecture 2 it's like a 2-star lecture. Not that important. However, for complementary sake, and if you really want to understand where NAD came from, you would go and see the lecture. Lecture number 3 is also a 5-star lecture. It's extremely important because basically we're taking biochemistry on metabolism, on the clinical significance, talking about disease called Pellagra and how the NAD plays a very big role in it. Okay? And of course, the vitamin deficiency associated with it. Now, with that being said, let us start with the NAD. So, the abbreviation NAD, remember how we did with NAD, with DNA and RNA? Always look at the weird people. So what does NAD stand for? Now, if you're watching these lectures in order, you already recognize what this molecule is, what this compound is, right? So basically, this is a ribose sugar. It has a phosphate, connected to a phosphate, and it has a nitrogenous base. Now, if you remember, the nitrogenous base, this is a purine. Why? Well, because it has 1 ring, 2 ring. What kind of purine is it? Remember, we said purines could be either adenine or guanine. Since it has an amino group on the carbon number 6 or the 12 o'clock position here, this is going to be adenine, right? Okay, great. What about this here? Well, it has a ribose sugar. It's connected to a phosphate group. And it would appear that this would be a base. And in fact, it is a base. Now, you might have not seen this base, but this base here is called nicotinamide. This is called nicotinamide. And so basically, why am I telling, oh yeah, by the way, a nicotinamide, this looks like a Q, wait, nicotinamide. A nicotinamide, right? So basically you're going to say, again, look at the word. It has something to do with nicotine, and you'd be absolutely correct. Basically, we take a nicotine, a cigarette, well, not a cigarette, nicotine, and you burn it. So you oxidize it, right? Of course, oxidation is not the same thing as burning. Basically, oxidation is controlled burning, right? But anyway, once you burn it, you would go from having a nicotine to having a structure that has, it's a 6-carbon ring structure, a pyridol, and it looks like this. What about the amide group? What's amide? What's nicotinamide? Now, I know this is an amino group, right? At this point, you should. So what's an amino group? What's the difference between an amide and an amino group? Listen, I'm going to say this really quickly. This right here, when the carbon is attached to a NH2, we call this an amino group. When the carbon is attached to an amino group, but is also attached to an oxygen, now this is called an amide group. And this is where we get the name nicotinamide. Okay? Now, so you still didn't explain the abbreviation. What's NAD? Where did it come from? N stands for nicotinamide, A stands for adenine. I don't know why I did that. Here's adenine, right? What about the D? Well, first of all, you're going to say, listen, dude, I mean, yeah, that's nicotinamide. This nitrogenous base is adenine. So why didn't you say this is an AMP? I mean, basically, this whole structure is AMP, and you'd be absolutely correct. Well, what about this structure? Why don't we call it simply nicotinamide monophosphate as well? And also, you could do that. Yeah, you'd be right. However, they said, listen, there's two nucleotides or dinucleotide, and one base is nicotinamide, the other base is adenine. So you know what? Nicotinamide, adenine, di meaning two, di means two, nucleotides. So there's the two nucleotides. Okay, this is what NAD is. NAD is nothing but an AMP, an adenomonophosphate, and a nicotinamide monophosphate or a nicotinic acid monophosphate that are linked to one another by a phosphodiester bridge. Remember, because there's a phosphate here, and these two structures are linked to one another. That's all it is. This is what nicotinamide comes to, okay? So recognize it, know it. When you see nicotinamide, I don't want you to know the structure. I just want you to understand what it is exactly, right? So it's nicotine and adenine, and they're nucleotides. Okay. Now, what exactly is NAD? Now, in the lecture about cofactors, remember, in the lecture about cofactors, we said that cofactors are subdivided into inorganic cofactors or organic cofactor. And we said that organic cofactors are also called coenzymes, right? Now, the inorganic cofactors, remember we said they're zinc, they're magnesium, they're copper, they're ferrous. The organic cofactors are going to be things like NAD. Right? So again, just really quickly, what is NAD, FAD, of course, tetrahydrofolate, something like that. Now, so what exactly is a cofactor? A cofactor is a structure that's not made out of protein, okay? So basically it's a chemical structure that helps an enzyme work, do its job. Now, NAD has two forms. Listen, so far we did definition, this. We did the classification, what class does it belong to? What about the forms? Listen, NAD comes in two forms, right? It can be in the oxidized form, and please do not be scared about the word oxidized or where it came from. Again, we have a lecture, if you saw the lecture on the redox reactions, on the dehydrogenase reactions, and specifically, you would understand what it is. I'm going to tell it to you really quickly. The other form is NADH. This is the reduced form. So NAD, this is an oxidized form, right? The NADH is going to be a reduced form. Basically, listen, we added the hydrogen to the NAD, we reduced it, okay? Because we added the hydrogen, because we added an electron, in fact, which is, okay, this gets me to another point. Okay, I'm going to talk about it in a moment, just bear with me. Because we added a hydrogen to the NAD, it went from the oxidized form and became a reduced form, right? Now, I'm not going to talk and explain about it further. You can look it up. We have a good lecture about it, extremely good lecture, in fact, right? You'll understand anything. Now, the hydrogen. Listen, a lot of students have a dilemma with hydrogen because sometimes we refer to it as a hydrogen atom, sometimes as a proton, sometimes some lecturers out there say a negative hydrogen ion, for instance, which is, to be honest, not really correct. Listen, hydrogen, and this is important, hydrogen comes in three forms. Now, if you have neutral hydrogen, remember the hydrogen has a proton in its nucleus, and on the outer shell there's going to be an electron. It's not important. Listen, what is important, if the hydrogen has a proton and has an electron, then it's neutral. So it's going to be like this, right? What do we call this form? We call this form a hydrogen atom. A-T-O-M, it's an atom. It's a hydrogen atom, right? Now, if the hydrogen has a proton but has no electron, then this is called a proton. This is called a proton, right? So we've got hydrogen atom, which is neutral. When it's positive, we call it proton. What about when it's negative? So basically, how does it become negative? It has two electrons. It has two electrons, it has one proton, and so the end result would be a hydrogen that is negatively charged. What do we call it? We call this a hydride, here, hydride ion. Okay? Now, is it important to understand these. It's absolutely essential to understand them, and you're going to see why when we talk about the dehydrogenase enzymes. And in fact, we're going to see some maybe examples about the NAD and how it works. Okay, so that is the form. Now, what is the function of NAD? What does it do? And so, you know, textbooks, Google, whatever, they say it's an electron courier. Okay, great, nice. What's an electron courier? What does it mean? Basically, NAD, you can think about it as an Uber, right? Now, electrons, biologically, cannot be simply, you know, floating around in the cells or in the human body. They do not do that. Instead, what they do is they attach themselves to a hydrogen, right? Now, listen, I'm going to make it easier. In the body, in the human body, we have two processes of metabolism. You have the catabolic process, and for any of you that's how the introductory lecture on biochemistry, you'd know this. So you've got catabolic, you've got the metabolic, right? Listen, catabolic, basically anything that has to do with lysis. So we're breaking stuff down, whether it be glycolysis, lipolysis, it could be, you know, the breakdown of alcohol, right? Ethanol metabolism, even in the Krebs cycle, right? So when we break things down, why do we break them down? What is the end result we want to reach? Well, basically, you want to produce or generate an adenosine triphosphate, an ATP, which we are going to use for energy. When we are doing the metabolic pathways, usually it's going to be something that has to do with genesis. Genesis is the suffix, meaning that we're building something, like gluconeogenesis, glycogenesis, lipogenesis, right? Basically, this is going to use ATP, right? Okay, great. So why am I talking about this here? Because NAD plus is going to be part of the catabolic pathway in most cases, right? So as a general rule of thumb, when you see something with lysis in it, know that the cofactor that is usually going to be used is going to be NAD plus. Why? Because when we destroy it, you do glycolysis, right? You do beta oxidation for fatty acids. You do alcohol metabolism. What are you doing? You're breaking them down. Okay, so, and they generate ATP. Well, not directly. What they generate are electrons. Of course, electrons are not going to be present by themselves. They're going to have to go and ride on that good neutral hydrogen, right? Which is the hydrogen atom. Now this hydrogen atom becomes something like this. So it has two electrons and one proton. It's a hydride ion, right? And the NAD is going to go and pick up this hydride ion and take it somewhere else, then somewhere else. Then we enter the mitochondria. Then, you know, it's the Krebs cycle, okay? And eventually, after many steps, you get an ATP. So basically, the ATP needs electrons. Where those electrons come from? They come from anything that has to do with catabolism. Okay, good. Let's remove this. What about metabolism or metabolic? We're going to talk about this when we talk about the NADPH, okay? Okay, listen. So we did the definition, we did the classification of it, we saw the forms, we saw the hydrogen forms. What else are we missing? Let's make sure we didn't miss anything. Okay. All right, listen. Now, before we switch to the tablet, the enzyme, when you hear the word dehydrogenase, by the way, dehydrogenase, there's a lot of enzymes that are going to use like, for instance, malate dehydrogenase, isocitrate dehydrogenase, there are many of them, lactate dehydrogenase. What does dehydrogenase means? It means to remove. What are you removing exactly? You're removing hydrogen. How many hydrogens? You're going to remove 2 hydrogens, right? Now, to get a better understanding of all of this, I'm going to show you on the tablet, on the whiteboard of ours. I have written down some reactions. I don't want you to memorize the formula. I do not want you to memorize the exact reactions, this chemical structure. I just want you to understand. I want your brain to get the blueprint of how NAD is going to be used as a cofactor in these reactions. Okay? So let's start. Listen. Now, before I tell you about the reactions, let me give you an idea about the general reaction that we can see when we're talking about the NAD, right? Basically, here's what's going to happen. NAD plus 2 hydrogen molecules, the end result is something that is going to look like this. NADH plus a proton, or H positive, right? Okay. to make things easier, listen. Imagine this is a substrate. Which kind of substrate? Nobody cares. It's a substrate. And there is a dehydrogenase enzyme acting on it, right? So let's call this dehydrogenase a substrate, referring to this substrate right here. So it's a substrate dehydrogenase, okay? And now what's going to happen is that this enzyme, the substrate dehydrogenase, is going to take 2 hydrogen atoms out of this substrate, right? So here we go. It's, of course, atoms, so they're neutral, and 2 of them, right? So proton and electron on both. Now, what's going to happen is that this substrate dehydrogenase enzyme, it's kind of a mental enzyme. So it doesn't like things to be balanced. It's kind of psychotic. It takes the electron on this hydrogen and transfers it to the electron on this. So basically, the end result would be we've got 1 hydrogen that has 1 positive or 1 proton and 2 electrons, and we call this a hydride, right? We call it, and we talked about it, we said this is a hydride, right? And the other hydrogen is going to be in, well, in the proton form. Okay, great. So now we've got a hydride ion, and we've got a proton. Where do we put them? Well, here's where the NAD comes in, right? Remember, NAD plus. So what's going to happen is that this one, the hydride ion, is going to be caught by the NAD plus, and it's going to be reduced to NADH. What about the proton? The proton is going to remain a proton and stay, free. Okay. So what happens is you get a product, and this product is going to, by the way, this product here is the same product, is the same thing as the substrate if we add 2 hydrogen to it. Okay. Not important. We'll talk about this when we do dehydrogenase. Now, that being said, let me show you a couple of examples on exactly what I'm talking about. Listen. Let's take the enzyme, for instance. Okay, let's change the color. Let's talk about the enzyme malate dehydrogenase, right? So you've got a malate here. So what do we care about the malate? Well, let's count how many hydrogens malate has. Here's hydrogen number 1. Here's hydrogen number 2. I mean, sure, it's attached to a hydroxyl group, but a hydrogen it is. Here's hydrogen number 3, and here's hydrogen number 4. So the malate dehydrogenase is, of course, called malate because the dehydrogenase enzyme is going to act on the malate, right? What happens is that this dehydrogenase enzyme is going to remove 2 hydrogens. It's going to remove this hydrogen here, and it's going to remove this hydrogen here. Remember, the dehydrogenase removes the hydrogen. And of course, we have a hydrogen, it has an electron, it has a proton, the same thing with this, an electron and a proton. Then the psychopath is going to take electrons and put them on this hydrogen here. Eventually, you get a hydride ion and you get a free proton, right? Okay, and that's great. So the hydride ion is going to come and it's going to be attached to the NAD. And so it gets reduced because remember, it has an electron, or it has 2 electrons, so we reduce it. Now it's NADH, and the proton is going to be thrown out into the fluid or whatever. Now, what's the product? What's the end result? The end result is something called oxaloacetate. How many hydrogens do we have? Well, we have 1, we have 2, and that's all we have. So basically, remember, malate, we had 4 hydrogens. But since the dehydrogenase enzymes removed 2, we're left with something called oxaloacetate, only 2 hydrogens, right? Okay, let's take another example. Okay, so glyceraldehyde 3-phosphate, right? Again, let us count the hydrogens. So you've got hydrogen number 1, you've got hydrogen 2, 3, and 4. So basically you have 4 hydrogens. Of course, the enzyme that's going to act on it is called glyceraldehyde 3-phosphate dehydrogenase, right? What's it going to do? It's going to take 2 hydrogens out of the glyceraldehyde, right? And again, the same process occurs. Eventually, we have a hydride ion and we have a proton. The hydride ion, of course, is going to go, it's going to link with the NAD plus, and of course, it's going to reduce it to NADH and have a free proton. Okay, what's the end result? The end result is called a 1,3-bisphosphoglycerol. Now, here we had 4 hydrogens and removed 2. So you would expect that we're going to have only 2 hydrogens, right? Technically, we should. However, here we had 5 hydrogens, not 4. So there's going to be 5 hydrogens. And here you would expect to see 3. But when we count it, we find 1, 2, 3, and 4. So where did that extra hydrogen come from? Basically, we also added a phosphate group here, and the phosphate group had a hydroxyl group on it, and that's where it came from. And that's why we end up with 4 hydrogen ions instead of 3. But basically, listen, don't get mixed up about, the phosphate and whatever. I mean, if we didn't add the phosphate here, basically, we would be left with 3 hydrogens. Okay, listen, let me give you another example. Isocitrate, for instance, to alpha ketoglutarate. Basically, the same thing. Isocitrate dehydrogenase, because the dehydrogenase is going to act on isocitrate. It takes, 2 hydrogens out of the isocitrate, hydride ion, and a proton, and eventually the hydride ion is going to go and link up with the NAD plus, right, and gives you NADH, and the free proton is going to leave as a free proton. Of course, listen, there is an intermediate here. Some stuff happen, and eventually get alpha ketoglutarate, and so if you count the hydrogen ions here it's only 1 less, but that's because of the other substrates. Listen, basically, any dehydrogenase enzyme is going to take out 2 hydrogens, stick one of those, the hydride ion that is, on the NAD, and the other one is going to be thrown out freely. Okay, that's all it is. Now, to give you also, because I'm going to give you a general picture, remember that dehydrogenase, most of the time it is associated with NAD, sometimes the cofactor is going to be FAD. Still does not change the way dehydrogenase works. On succinate, so with enzyme succinate dehydrogenase is going to remove 2 hydrogens, right? So here it has 2 hydrogens here, 2 hydrogens here, so it has a total of 4 hydrogens, which are going to go and bind to the FAD and give the FADH2. So the fumarate is the product, right? What's the difference between the fumarate and the succinate? Well, we removed 2 hydrogens. We removed 1 from here and 1 from here. And so we are left with 2 hydrogens. This is called fumarate. So if I add another 2 hydrogens to fumarate, guess what I'm going to get? I'm going to get succinate. Okay?